Imagine a cold case, maybe decades old, suddenly cracking wide open. Or someone wrongly convicted finally walking free. Or the huge task of identifying victims after a disaster.
Yeah, and behind many of those stories today, there's this incredibly powerful technology, DNA forensics. It offers a kind of precision that, well, we used to seem like pure science fiction.
It really did. So maybe let's back up. Forensic science itself, what are we talking about fundamentally?
Fundamentally, it's just using science and technology to answer questions in legal cases, both criminal and civil. Before the mid-1980s, the toolkit was, well, different. Some tools were pretty solid, fingerprints, blood typing.
Reliable stuff.
Right. But others, like bite mark comparisons or looking at hairs under a microscope, turned out to be not nearly as reliable as people once thought.
Okay, so that sets the stage. Then comes this massive shift, 1986, you said, the first forensic use of DNA profiling.
Exactly. And it just completely transformed how police identify biological evidence. Suddenly you could get DNA profiles from, unbelievably small things. Saliva on a cigarette butt, a pet hair, a tiny, tiny blood stain.
Like pinhead-sized?
Yeah, literally. And it wasn't just about pristine samples either. Even stuff that's been degraded, by fire or just time passing can still yield a usable profile.
Which is huge for proving innocence or guilt or just giving answers. You mentioned identifying victims from mass disasters.
Absolutely. Think about the 2004 Asian tsunami or the aftermath of 9/11. DNA was crucial. And it's even used for things like identifying endangered species or tracking trafficked animals. It's surprisingly versatile.
And it goes way beyond crime scenes too, right? Paternity tests, identifying plants, even confirming military casualties.
Evolutionary studies, yeah. It's become a really foundational tool in biology.
So our mission today in this deep dive is really to get into how DNA profiling actually works, right? How do we interpret the results? What's the deal with these DNA databases? What are the problems, the ethical stuff, and maybe where it's all headed?
That's the plan, to really unpack this incredible leap in scientific investigation.
Okay, let's do it. To understand this revolution, we probably need to go back to the beginning. Where did it all start?
It really kicked off with Dr. Alec Jeffreys. He published this landmark paper back in 1985. He was looking at something he called mini-satellites.
Mini-satellites.
Yeah, or more formally, variable number tandem repeats, VNTRs.
Right, VNTR. So these are like repeating bits of DNA. How did he figure out they could identify people?
Well, he found that these sections, there may be 15 to 100 base pairs long, but they repeat over and over again. And the key thing was the number of times they repeat varies a lot between different people.
Oh, okay. So the length of that whole VNTR section is different for you versus me.
Exactly. Like maybe you have a 16 base pair sequence repeated 3 times, but I have it repeated 20 times. That difference in length becomes a unique marker.
And that variability, that must be the key to its power.
Totally. Because for any single VNTR spot, there might be, say, up to 30 different possible lengths or alleles floating around in the population. So if you just look at 4 different VNTR locations, and each has maybe 20 possible alleles.
The number of combinations explodes.
It explodes. You're talking about like 2 billion possible genetic combinations just from those 4 spots. That's massive discriminatory power.
Okay, so how did they actually do it back then? How did they see this pattern?
It was quite a process. First, extract the DNA, then use these special enzymes, restriction enzymes, to cut the DNA on either side of the VNTRs.
Okay.
Then you separate those fragments by size using gel electrophoresis. The smaller pieces move faster through the gel, the bigger ones slower.
Right, I remember that from biology class.
Yeah. Then they transfer that pattern onto a membrane and use a radioactive probe that sticks only to the VNTR sequences. When you expose it to X-ray film, you see this unique pattern of bands.
Like a barcode.
Kind of like a genetic barcode, yeah.
Right.
Larger VNTRs near the top, smaller ones near the bottom. And that pattern was unique to the individual, no matter where the DNA came from, blood, semen, whatever. Scientists usually looked at about 5 or 6 of these locations to build a profile.
But you mentioned limitations. This early method wasn't perfect for every crime scene.
Definitely not. The big one was the amount of DNA you needed. Something like 10,000 cells or 50 milligrams of DNA.
That sounds like quite a lot.
It is, especially for a crime scene where you might only find trace amounts. So VNTRs were used a lot for paternity testing, where getting the big sample is easy. But for forensics, it's mostly been replaced by newer, more sensitive techniques.
Which brings us to maybe the most famous early case, the Pitchfork case. That story is just incredible.
It really is. It happened in Leicestershire, England, mid-80s. 2 young women, Linda Mann and Dawn Ashworth, were raped and murdered. Police had very few leads. They eventually questioned the local guy, Richard Buckland.
And he confessed.
He confessed to the second murder, Don Ashworth's. But there were doubts, especially about the first murder. So the police went to Dr. Jeffries. This was 1986. They asked him to use his new VNTR technique on semen samples from both crime scenes.
Yeah.
His analysis showed first that the same man committed both crimes. Huge. But then, the absolute bombshell. The DNA profile from the crime scenes did not match Richard Buckland.
Wow. So their only suspect was innocent.
Exactly, which led to something totally unprecedented, the first ever mass DNA screening. They asked basically every adult man in the area to voluntarily give a blood or saliva sample.
How many people are we talking?
About 4,000 men came forward. It was massive.
Did it work?
Eventually, yes, but not directly. Turns out, a man named Colin Pitchfork, who worked in a local bakery, had persuaded a friend to give a sample for him, using a fake ID.
No way.
Yeah. But someone overheard the friend bragging about it in a pub, reported it to the police. Pitchfork was arrested, his DNA was taken, and he was a perfect match to both crime scenes.
Unbelievable.
He confessed, got life in prison, and crucially, Richard Buckland, the man who falsely confessed, was completely exonerated. It was the first time DNA profiling solved a crime, and the first time it proved someone innocent. Just a landmark case.
That really drives home the impact. But as you said, the technology needed to get more sensitive. That sample size issue was still a problem.
It was. And the next massive leap forward was the invention of the polymerase chain reaction, or PCR.
PCR. Right, that's the technique that makes lots of copies of DNA.
Exactly. It's like a genetic photocopier. You can take a tiny amount of DNA and amplify specific regions millions, even billions of times in just a few hours. Uses specific little starter sequences called primers and a special heat-stable enzyme.
So suddenly you didn't need that big 50-milligram sample anymore.
Not at all. PCR meant you could work with incredibly small amounts. A single hair root, a few skin cells under fingernails, even DNA from really old or degraded samples, think ancient bones, Egyptian mummies.
Wow. Okay, so PCR makes tiny samples usable. How did that change the type of DNA markers used?
It allowed a shift to something called short tandem repeats, or STRs. These are similar to VNTRs, but the repeating units are much shorter, usually just 2 to 9 base pairs long.
Shorter repeats, okay.
Yeah, and they repeat maybe 7 to 40 times. So, for example, a common STR location called D8S Lun179 has the sequence TCTA repeating anywhere from 7 to 20 times. That gives you 19 possible alleles just for that one spot.
And because they're shorter, they're easier to amplify with PCR, especially from degraded DNA.
Precisely. The smaller size makes them much more robust for PCR amplification, even if the DNA is broken down. That's why STRs are the standard in forensics today. They work reliably with the kind of trace evidence you actually find.
And is there standardization now? Like, does everyone look at the same STR locations?
Yes, thankfully. There are hundreds of STRs known, but forensic labs use a standardized set. In the U.S., the FBI uses a core set of 20 STR loci. Most European countries use 12. This standardization is crucial for comparing profiles and sharing data between labs and databases like CODIS.
Okay, so let's walk through the modern process. How do they actually analyze these STRs now? It sounds much faster than the old radioactive probes.
Oh, way faster and much more automated. They use commercial kits that contain primers for all the core STR loci. And here's the clever part. The primers for different loci are tagged with different colored fluorescent dyes.
Different colors for different locations.
Right. Or sometimes different locations get the same color, but the size of the DNA fragment produced will be different because the number of repeats varies. So you can distinguish them by color and size.
And how do they measure the size and color?
And that's done using capillary electrophoresis. Instead of a big flat gel, the amplified DNA fragments are run through these really thin glass tubes, like capillaries, filled with a gel-like substance. An electric current pulls the negatively charged DNA through the tube. Shorter fragments sip through faster, longer ones lag behind. At the end of the tube, a laser shines through, and a detector picks up the fluorescent signal from each tagged fragment as it passes.
And that creates the profile.
Exactly. The software analyzes the data and generates a graph called an electropherogram. You see peaks representing the alleles. If you inherited different numbers of repeats from your parents for a particular STR, heterozygous, you'll see 2 peaks. If you got the same number from both, homozygous, you see 1 taller peak.
And the position of the peak tells you the size, which tells you the number of repeats.
You got it. The final STR profile is basically just a list of the number of repeats for each allele at each of the core loci. And modern systems can analyze dozens of samples really quickly, maybe in under an hour.
That efficiency is incredible. Now, besides these standard STRs, are there specialized methods for trickier situations? You mentioned Y chromosome STRs.
Yes, YSTR profiling is really important. It specifically looks at STRs found only on the Y chromosome.
Which only males have.
Right, so it's invaluable in cases with mixed male-female DNA, like sexual assault evidence.
Mhm.
It lets you isolate and profile just the male contributor. It's also useful if you have multiple male suspects mixed together.
Makes sense. But the Y chromosome has that direct father-to-son inheritance, doesn't it? Does that cause issues?
It does create limitations. Because it's passed down largely unchanged, YSTR profiles can't distinguish between fathers and sons or brothers or any males related through an unbroken paternal line. Even distant male relatives might share the same YSTR profile.
So it's good for isolating male DNA, but not always great for telling specific males apart if they're related.
Exactly. But it's still very useful for things like identifying missing persons if you have a paternal relative's sample for comparison, or for tracing male lineages in genealogy.
Okay. What about mitochondrial DNA, mtDNA?
mtDNA is another crucial tool, especially for really challenging samples.
Because there's lots of it in each cell.
Loads. Each cell has hundreds or even thousands of mitochondria, and each mitochondrion has its own little circular DNA chromosome. This high copy number makes mtDNA analysis fantastic for samples that are very old, very degraded, or just tiny like hair shafts without the root, or old bone fragments.
And it has a different inheritance pattern too, right? Not like the chromosomes in the nucleus.
Completely different. You inherit your mtDNA almost exclusively from your mother, through the egg cell. It's passed down the maternal line, basically unchanged, without recombination.
So how is it analyzed?
They amplify specific variable regions within the mitochondrial DNA loop, sequence those regions, and then compare the sequence to reference samples.
Which makes it perfect for identifying victims of mass disasters, like you mentioned earlier, if you can get a sample from a maternal relative.
Precisely. It was vital after events like the Srebrenica massacre and the World Trade Center attacks. But, like YSTRs, it has limitations for individual identification. All siblings, and anyone related through the maternal line, will share the same mtDNA profile. It's also heavily used in evolutionary studies and tracking human migrations.
Right. Okay, one more type. SNPs. Single nucleotide polymorphisms. How do they fit in?
SNPs are actually the most common type of genetic variation. They're just single base pair differences at specific points in the genome. A C might be changed to a T, for example, or a small insertion or deletion. There are millions of them.
Millions sounds powerful. Why aren't they the main tool for forensics instead of STRs?
Well, the thing is, most SNPs only have two common variations, or alleles. STRs, with all their different repeat numbers, are much more variable at a single location. So to get the same power to tell people apart as you get with the 20-core STRs, you need to analyze a lot more SNPs, maybe 50 or more.
I see. So STRs are more efficient for discriminating between individuals. What's the advantage of SNPs then?
Their biggest advantage is for really degraded DNA. Because you're just looking for a single base change, the piece of DNA you need to successfully amplify using PCR can be incredibly short, maybe only 50 nucleotides long.
Ah, so even if the DNA is badly broken down, you might still get SNP information when STRs fail.
Exactly. That makes them potentially very useful for those extremely challenging samples. But for routine forensic casework, STRs are still the workhorse. SNP analysis is more common right now for ancestry testing or analyzing Y chromosome and mtDNA variations for lineage studies.
Okay, now let's touch on something that sounds pretty futuristic and maybe a bit controversial. DNA phenotyping.
Yeah, this is definitely cutting-edge stuff. And it's important to understand the difference. Regular DNA profiling aims to match a sample to a known individual. DNA phenotyping tries to predict what an unknown person might look like or where their ancestors came from based just on their DNA.
Predict what they look like. Seriously, how accurately?
Well, for certain traits, surprisingly accurately. Things like eye color, using just a handful of SNPs, they can predict blue versus brown eyes with something like 95% accuracy. Hair color, black, brown, red, and skin pigmentation can also be predicted with pretty high accuracy, maybe 80–90%, using specific sets of SNPs. Determining biological sex and broad geographic ancestry is also quite reliable.
That's already quite specific, but I've seen reports claiming they can predict actual facial structure, like generate a sketch from DNA.
That's where it gets more controversial and, frankly, less scientifically validated right now. Some companies and researchers claim they can do this, creating 3D facial models or even photo-like images. But the science linking specific genes to complex facial features is still very much developing. It's not nearly as established as predicting eye or hair color.
So can police use this in court?
Not really. Not yet for identification. It hasn't met the scientific validation standards for courtroom evidence regarding identity. But law enforcement is using it, sometimes calling it forensic intelligence. They might use a predicted profile, say, likely male, Northern European ancestry, brown hair, blue eyes, to help narrow down suspect pools in cold cases, or to help identify unknown remains. It's an investigative lead generator, not proof of identity.
Fascinating, but definitely raises some questions. Okay, so let's say we have a standard STR profile from a crime scene and one from a suspect. What does it mean if they match?
That's a really important question. If the profiles don't match, it's simple. The suspect is excluded. They could not have left that sample.
Clear cut.
Right. But if they do match, it's more complex. It could be the same person, or it could be two different people who just happen to share that same DNA profile by chance.
Ah, okay. So how do you express the significance of that match?
That's where statistics come in. We calculate something called the profile probability, or more commonly, the random match probability, RMP. This tells you the estimated frequency of that specific DNA profile in a given population.
So, like, how likely is it that a random unrelated person would have this same profile?
Exactly. And it's calculated using population allele frequencies and the product rule.
Okay, break that down. How does it work?
So for each STR locus, we know how common each allele, each repeat number is in various reference populations. Let's say at one locus, the suspect has alleles 11 and 13. We look up the frequencies of allele 11, let's call it P, and allele 13, Q, in the relevant population database. The probability of having that specific heterozygous combination, 11-13, is calculated as 2 times P times Q.
Okay, 2PQ for heterozygotes. What about homozygotes? Same allele from both parents.
If they have 2 copies of allele 11, the probability...
You calculate that probability for each locus in the profile.
And then you use the product rule. You multiply the probabilities for all the independent loci together.
And because you're multiplying probabilities, which are less than 1, the overall number gets smaller and smaller with each locus added.
Exactly. A single locus might be shared by, say, 10% of the population. But when you multiply the probabilities across 13, or even better, the 20 core STR loci.
The final random match probability becomes incredibly tiny.
Astronomically tiny. For a full 20 locus STR profile, the RMP can be rarer than 1 in a trillion, often much rarer, like 1 in sextillions or octillions.
So, for all practical purposes, does that mean a full STR profile is unique to an individual?
With a very high degree of scientific certainty, yes. That incredibly low RMP means the chance that another unrelated person chosen at random would share that exact profile is virtually zero.
Virtually zero, but not absolutely zero. Are there exceptions?
Yes, crucial exceptions. The most obvious one is identical twins. They develop from the same fertilized egg, so they have identical DNA and will have identical STR profiles.
Okay, twins are the same. What about other relatives?
Siblings share, on average, 50% of their DNA. So they have a much higher chance of sharing alleles at any given locus compared to unrelated people. They might share one allele, about 50% chance, or even both alleles, about 25% chance at a locus. Other close relatives also share more alleles than random individuals.
So, if the suspect has a brother, the standard RMP calculation doesn't quite apply.
Correct. Forensic labs often perform additional calculations to estimate the probability that a close relative, like a sibling, might share the profile. Also, standard allele frequency databases assume large, randomly mating populations. If the sample comes from a small, isolated, or highly interrelated group, those standard frequencies might not be accurate, and the RMP needs careful consideration.
That makes sense. And a huge part of modern DNA forensics relies on comparing profiles to databases, right? Like CODIS in the US.
Absolutely. Databases are essential. The UK set up the first national DNA database back in 1995. CODIS, the combined DNA index system run by the FBI, is the US equivalent. As of a few years ago, it held over 17 million profiles.
17 million. What kinds of profiles are in there?
It's mainly divided into 2 parts. The offender index contains profiles from people convicted of certain crimes. The laws vary by state. And the forensic index contains profiles developed from crime scene evidence where the source is unknown.
So you can compare crime scene DNA to convicted offenders.
Right. Or compare DNA from different crime scenes to see if they're linked. Some states also allow profiles from arrestees. people arrested but not yet convicted, missing persons, and unidentified human remains to be included.
The value seems undeniable when you hear about cases solved or wrongful convictions overturned. The Kennedy Brewer case you mentioned earlier sounds like a prime example.
It really is. Kennedy Brewer was convicted in Mississippi in 1992, mainly based on bite mark evidence that was later shown to be totally flawed. DNA testing at the time wasn't sensitive enough. He was sentenced to death.
Based on bite marks, that's chilling.
It is. Years later, in 2001, improved DNA testing on the original evidence excluded Brewer. It also excluded his male relatives. But incredibly, he still wasn't released.
What? Why not?
It took the Innocence Project getting involved in 2007. They pushed for further testing and for the profile to be run through CODIS. And bingo, it matched another man, Justin Albert Johnson, who had been an early suspect and had a history of assaults. Johnson then confessed.
And Brewer was finally exonerated.
Finally, yes. And it gets even crazier. Johnson also confessed to another murder, one for which a different man, Levon Brooks, had been convicted, again based on the same discredited bite mark expert. Brooks was exonerated too.
Wow. 2 exonerations linked to one CODIS hit and flawed bite mark testimony. That's powerful.
Absolutely. And these aren't isolated incidents. Since 1989, DNA has led to over 350 exonerations in the U.S., including 17 people on death row. And in over 150 of those cases, the actual perpetrator was identified, often through CODIS.
The impact on justice is clear. But storing millions of people's genetic blueprints must raise some concerns, too.
Huge concerns. Privacy, civil liberties, the potential for misuse, who gets put into the database, under what circumstances. These are massive societal debates balancing public safety against individual rights.
Definitely a tension there. And even putting aside the database ethics, the technology itself isn't foolproof, is it? There are limitations and potential errors.
Oh, absolutely. For one thing, many crimes simply don't involve recoverable DNA evidence, or the DNA might be too degraded or contaminated to get a usable profile. Labs also face huge backlogs, valuable evidence that's waiting to be tested.
And human error, mixing up samples.
It happens. Mistakes in collection, labeling, or analysis have led to wrongful convictions. It's rare, but devastating when it occurs. Then there's the complexity of interpreting mixed samples DNA from multiple people combined, or dealing with partial profiles from degraded DNA. That can be really challenging.
You mentioned transference earlier, too. The Lucas Anderson case sounds like a cautionary tale about that.
It's an absolutely mind-blowing case. 2012, a wealthy man, Ravish Kumra, is murdered. DNA from under his fingernails is a perfect match to Lucas Anderson, a homeless man with a criminal record. Anderson is arrested, charged with murder, looking at the death penalty.
Seems like an open-and-shut case based on the DNA.
Except Anderson had an absolutely rock-solid alibi. At the exact time of the murder, he was extremely intoxicated and unconscious in a hospital emergency room being treated by paramedics. Records proved it. He couldn't possibly have been at the crime scene.
So, how did his DNA get under the victim's fingernails?
That baffled everyone for months. Eventually, investigators figured it out. The same paramedics who treated Anderson at the hospital later responded to the 911 call at Kumra's house after the murder.
Oh, no.
Yeah. Somehow, Anderson's DNA, maybe from his skin cells on their equipment or clothes or gloves, got transferred by the paramedics onto Kumra's body during their response at the crime scene.
That is incredible and terrifying. If he hadn't had that perfect hospital alibi.
He could easily have been convicted, possibly executed, based on seemingly incontrovertible DNA evidence. It just highlights how sensitive DNA detection is, how easily it can be transferred, and the danger of relying only on DNA without considering all the other evidence and context.
A really stark reminder. And if accidental transference is possible, what about deliberate planting of evidence?
That's another serious concern, especially as DNA synthesis technology gets better. Criminals could potentially plant biological material hair, saliva at a scene to frame someone,
Or even create fake DNA.
There was research published back in 2010 showing it was possible to synthesize DNA fragments matching a known STR profile, mix them with, say, saliva, and plant that sample. Routine forensic tests couldn't distinguish it from real DNA.
Yikes. So forensic labs need ways to detect synthetic DNA now.
It's becoming a potential need. One idea is looking for epigenetic markers, like DNA methylation patterns, which are present on natural DNA, but wouldn't typically be on synthetically created DNA. It's an ongoing challenge.
So stepping back, all this powerful tech forces us to grapple with some big ethical questions, doesn't it?
For sure. Like we touched on with databases, who should be in them? Should police be allowed to collect abandoned DNA, say from a discarded coffee cup, without consent?
Right. And what about familial searching? searching databases not for a perfect match, but for partial matches that might indicate a relative of the perpetrator.
That's hugely controversial. It essentially brings entire families under genetic surveillance based on the actions of one relative. Is that scientifically sound? Is it ethical? Different jurisdictions have very different rules about it.
And DNA phenotyping, again, using DNA to predict appearance or ancestry. Using that to generate leads or even identify suspects raises all sorts of ethical flags about potential bias and the reliability of the predictions.
Exactly. These aren't easy questions. As the technology gets more powerful, the ethical considerations just become more complex and more urgent. We need to constantly evaluate the balance between solving crimes and protecting fundamental rights.
We have certainly covered a lot of ground today. We've gone from the very beginnings of DNA forensics with Dr. Jeffries and those early VNTRs through the PCR revolution, the rise of STRs.
Specialized techniques like YSTR and mtDNA, the power of databases like CODIS, and even peered into the future with phenotyping and the challenges of synthetic DNA.
It's clear this technology has incredible power. It solved countless crimes, brought closure to families, exonerated hundreds of innocent people. Just amazing.
Truly remarkable. But, as we've seen, it's not magic. It has limitations. Degraded samples, mixtures, the potential for error, for transference, even for deliberate tampering.
And those profound ethical dilemmas surrounding privacy, data collection, familial searching, and how we use predictive information.
They are absolutely central to the conversation.
So if we connect this to the bigger picture, looking ahead, how do we navigate this? How do we maximize the potential of DNA for justice and truth while fiercely protecting individual rights and guarding against misuse or over-reliance? What safeguards do we need as this technology inevitably becomes even more powerful?
That's the critical question we need to keep asking, isn't it? It requires ongoing dialogue, careful oversight, and a commitment to both scientific rigor and ethical principles.
A lot for us all to think about. Well, thank you for joining us on this deep dive into the truly fascinating, complex, and ever-evolving world of DNA forensics.